cost effective because it must be cheaper than other methods like the microRNA sequencing. Because if you want to use the microRNA sequencing, you need to a lot of the machines or lab requirements to do that. So about the disadvantage is must be the limited dynamic range and cross hybridization and require period knowledge. Because the microRNA.
Okay, sorry, Jianto, you have 1 minute to go to sum up your presentation.
Okay, but in my view, there are two different methods can improve the microcrad ability. Is the LNA and TLDA. TLDA is based on the microfluid. That means you can use the a few samples to do a lot of analysis. Most of them be 384 wells. And the LNA can increase the accuracy in the microarrays. Because when they use in the PCR, it can detect the 1p ground the microRNAs in the PCR.
Okay, you please summarize in this slide your final word.
Summary. Sorry. Summary, they prove the those two microRNA can guide the radio expression of those daughters.
Okay then.
Yeah,
okay, thank you. Thank you so much for your presentation. Okay, a bit of heads up, for the next presenter. Please only focus on the method that you have given to present. Like if you are given a method on microarray, you present only on the microarray and on the validation. What did you do for the validation? You don't have to present everything about the article. Okay?
Oh, okay.
Okay, so can you please go to your slide advantage and disadvantage? Okay, you say it's limited dynamic range. It may not be able to detect a low abundance microRNA. So what is your suggestion to detect low abundance microRNA?
You mean low concentration?
What do you mean by here? What do you mean by this sentence? It may not be able to detect low abundance microRNA. What does it mean?
Because
okay.
Because you have not too much sample, you can not have enough the microRNA to detect. So it may be not too much the signals to be detected.
Okay, low abundance means low availability. Okay, low availability means the expression level of the microRNA is low. Okay, so you have to know, if let's say this is the case, this is the advantage of your method. What are the method that you can use to detect the low abundance microRNA? Okay. Okay, then never mind. We hear the next presenter. Thank you so much for your presentation.
Thank you.
So can we go to the next presenter? Next presenter, please present only the method I have given you, and what are the advantage, disadvantage, and what are the validation method that they use to validate the biomarker that they discovered. Okay, you don't have to present everything about the paper, and please don't present anything about the result. Here it's not, we do not want to hear result. We just want to know the methodology of the discovery. If you have prepared results in your slide, you can skip the result part. Focus on the methodology, the discovery part, okay? Next
Just
presenter.
yeah, when I prepared my slide, I prepared simultaneously, Doctor, the method and the result. Can I present it?
Don't have to present the result. We are not here to listen about the result for the paper. It's not significant for us. We want to know about the discovery, the methodology part, how they use the platform to discover the biomarker. You have to present on that.
Okay,
Okay.
noted, Doctor. All right.
Kavita, do you want to go next?
Can, Doctor.
Okay, please share your slide. The rest, please mute your mic. Okay, thank you.
So a very good evening. My name is Kavita, and the topic, the platform that was given to me was NanoString. And based on that platform, I managed to find a title. My title for today is Prognostic Biomarker Test for Triple Negative Breast Cancer Recurrence. Everyone is able to see my slide?
Yes, sure. Please proceed.
Okay. So what is triple negative breast cancer? This is an invasive type of breast cancer. It is described because of the absence of estrogen receptor, progesterone receptor, and HER2. Currently, the treatment is with aggressive chemotherapy, which can cause permanent peripheral neuropathy, cardiac toxicity, and secondary malignancy. As we can see here on the chart, the recurrence rate in the patients who had triple negative breast cancer, 58% had a disease-free survival. More than 8 years, they didn't get a relapse, whereas 42% had a relapse. So the researchers were wondering what is the difference between the relapse and the one who had the disease-free survival. When they looked into it, they were trying to figure out could there be a molecular approach to it. So they noticed that there was an abnormal signal pathway leading to immune resistance in triple negative breast cancer. This is an image from Zhang et al. In 2023. It shows that there's a signal pathway which leads to MHC transcription. MHC class 2 antigen presentation pathway in TNBC tumors was the most significant pathway associated with patients who relapsed compared with those who did not experience relapse. So they wanted to find a clinical scale assay to specifically measure the expression of the MHC2 pathway signature genes in clinical specimen. And what they come up, the conclusion was to either a qPCR assay or a nanostring assay that is compatible with a fragmented RNA derived from formalin-fixed paraffin-embedded clinical specimens could be a promising approach to determine if this discovery has clinical utility as a prognostic biomarker in larger case series of TNBC patients. So what is nanostring? Nanostring is better known as nanostring encounter. It is a direct digital detection of transcriptome. It can be quantified up to 800 genes. Basically, it comprises of these three major steps, which is the first step, hybridization. Nanostring has a pairs of reporter and capture probes. The reporter, as you can see in the diagram, is the colored beads. So it gives the signal, whereas the capture probe, the silver bead, is the probe that is used for immobilization. So this color-coded balls is actually the molecular barcode. As you can see in the diagram, the reporter and capture probes binds to the target genes, and then it will be proceeded for the next step. Next is the purification and immobilization. The excess probes will be removed and the complex probe target will bound, immobilize, and align to the plate. Then the counting and analysis will be done. This can be done in the nCounter digital analyzer. So based on this approach, I found a journal titled A Multigene Assay Determines Risk of Recurrence in Patients with Triple Negative Breast Cancer by Stewart in 2019. They have come up with the MHC2 immune activation assay design using NanoString 36 gene expression panel, which comprises of 13 genes in the MHC class 2, 9 tumor infiltrating lymphocyte, 9 subtype verification genes, and 5 housekeeping control genes for normalization. As we can see here, the pre-analytical testing, concordance between matched frozen and FFPE breast tumors was done, followed by technical replication in FFPE tumors, TIL gene detection in TNBC tumors, and the subtype verification gene threshold for inclusion criteria for basal-like TNBC tumors were done. When we see here, we can see that there's the basal-like HER2-enriched luminal A and luminal B was done. And this is the testing that was done using the NanoString. First, they wanted to compare whether the NanoString expression code concordant with the RNA sequencing. And they had a concordant result. The association of MHC2 score with the relapse status and the disease-free survival patient was done to see that there are more expressions of MHC2 and TIL genes in the non-relapse patient compared to the relapse patient. And finally, they were able to come up with the association of immune activation score with the relapse and non-relapse. As we can see here, the activation score is lower for relapse and higher for the non-relapse. So this was the validation of these studies was done in an independent population of 56 TNB stage 1 to 3 primary tumors. And they also managed to reproduce the same results where the activation score was lower for relapse patient compared to the non-relapse patient. And based on the NanoString result, the concordance of MHC2 expression was concordant with the MHC2 protein staining. When we see here, the MHC2 and TIL gene was higher in the non-relapse patient compared to the relapse. And finally, they're able to relate the TIL score with the histologic stromal TIL percent. So based on this, a validation of a threshold to identify TNBC patient with low risk of relapse, selection of an immune activation score threshold with 95% specificity in training set the experiment itself, whereas there was 100% specificity in the validation cohort. So risk of relapse is a linear function of the long transformed immune activation score. That means the higher the activation score, the lower the risk for patients to relapse. So as a summary, the preliminary data supporting potential clinical application. Patients with high immune activation score may have good prognosis whether or not they receive chemotherapy. So patients with high immune activation score may be more likely to respond to immunotherapy. So this is my advantages and disadvantages slide. The advantages of NanoString is it is a single tube multiplexing. So simultaneously, it can measure up to 800 RNA targets, which is much more higher compared to microarray. So we can have a comprehensive view of the gene expression profile. It is highly reproducible because there's no amplification or enzymatic step involved, as we can see in the qPCR technique.
Kavita, you have 1 more minute to go.
Noted, doctor. This is my last slide. And there's no reverse transcription. It is also a simple protocol with high sensitivity and specificity. It has a flexible sample input. That means a small amount of input material is sufficient enough to run this assay. When we see the previous slide, microarray could not perform with a small micro RNA samples, right? So we can tackle that with using NanoString. And finally, it has a robust performance because fresh, frozen, and formalin fixed paraffin embedded samples can be used. But there are definitely some disadvantages because it is limited genome coverage, not as big as the RNA sequencing. It's also a bit expensive and it's difficult to customize. Although it's simple, but to customize a custom panel is a bit challenging and difficult. And there are several other problems will arise if you are dealing with a unique experiment, which are the probe design optimization. And finally, it also has a limited dynamic range, and we can only find the sequence of nucleotides. So single nucleotide resolution is not available. So this nanostring is not applicable for SNPs or specific sequence variation. And this is my reference, my main one, and the others. That's all. Thank you.
Thank you so much, Kavita. Is there any question from the floor? Anyone wants to ask question? Any query? No one? Okay, Kavita. Why nanostring can only quantify up to 800 genes? Why 800? Why not 900?
Because it already has a pre-designed template which can only give up to 800 RNA targets. The total number of bells itself is 800.
So if you have more targets that you want to quantify, what you should do?
I will probably go to RNA sequencing.
Okay, then good. Okay, good presentation, Kavita.
Thank you, Doctor.
Okay.
So I want to
We
go
go to
next.
we go to the next presenter.
I want to go next, Doctor.
Okay, sure. Okay, even though you presented first, you have to remain in this presentation until all of your friends finish complete their presentation.
Yeah, no problem.
Leave early, okay? Okay.
Okay. Hello, can everyone hear me? Can everyone see the slide?
Yeah, sure.
Yeah. Okay. So, first of all, I'm Gian. Okay. So, do you sometimes feel like you're having dementia? Because sometimes I really tend to forget something. So sometimes I feel like, do I have dementia? So World Health Organization actually define what is dementia. It's the term of several diseases that affect memory, thinking, and ability to perform daily activities. And to date, there are 55 million cases of dementia worldwide, with more 10 million cases of new dementia cases being reported annually, and more 60 to 70% of them are comprised of Alzheimer's disease. So Alzheimer's disease is a disease of a neurological disorder in which they have categorized into three main stages, which are subjective cognitive decline, amnestic mild cognitive impairment, and lastly, it will progress into an Alzheimer's disease. However, to date, there is no cure available, and early detection of Alzheimer's disease is very important as it can delay the development of this disease and allow time to assess for treatment for symptom management. So it is important to for early diagnosis of Alzheimer's disease. Hence, this leads to this topic that I'll be sharing today, which is the scientist Zheng and her co-workers, which use circular RNA with a microarray platform to detect early onset of Alzheimer's disease. So the methods are shown here. They go through sample collection, microarray analysis. They annotate the interaction of circular RNA and microRNA, validate it using a real-time qPCR, construct the circular RNA, miRNA, and mRNA network. Then they look at the gene ontology pathway, and lastly, validate it with luciferase reporter assay. So the samples include 6 samples with 2 negative control, 3 SCD, and 1 AMCI samples from patients. And and then they perform microarray profiling of the circular RNA, in which they scrutinize this circular RNA based on the fold change of more than 1.5 and p-value of 0.05. Means they actually check on the differential expression of circular RNA in normal individuals as compared to that of the SCD and AMDI patients. So what is microarray profiling? So what happened is that initially you have to start extracting your total RNA from your blood samples, and then you have to add your RNAs to remove the linear RNA, because now we are detecting circular RNA. So circular RNA are very stable, and it is resisting RNA degradation due to their circular construct. Hence, when you add RNAs, it can remove linear RNA but enrich circular RNA. So in this case, you have to do cDNA synthesis for microarray profiling, and this cDNA will be labeled with fluorescence, and this labeled cDNA will be hybridized on the microarray chip. And then there are several advantages and disadvantages of microarray, as it is fast, the detection is fast, high throughput, and the analysis is simple and relatively low price as we compare it to the RNA sequencing. And however, there are several disadvantages as it relies on hybridization. It can be non-specific and it can be less accurate, causing a false positive result. And the analysis on microarray only allows analysis on predefined sequence because you need the probe on the chip. Okay, that's all the advantages and disadvantages. So what happened to this study is that they found out all these circular RNA through microarray profiling, which are differentially expressed in patients as compared to the normal individuals. And they have performed cross-validation of this differential expressed circular RNA as shown in the previous slide. So these 10 circular RNA are cross-validated with real-time qPCR, in which they have checked on the fold change based on the CyberGreen assay of qRT-PCR. And then they found out that these two circular RNA, which are 1481 and 479, are significantly upregulated in patients as compared to negative control. So these are the summary of the methods. Okay, so in brief, circular RNA inhibits miRNA and niRNA inhibits mRNA translation. So circular RNA indirectly actually regulates our gene expression by sponging miRNA expression. So they also performed luciferase reporter assay in which they found out that circular RNA can significantly reduce expression of this miRNA regulating the gene of EMB, and this 00479 circular RNA significantly reduce expression of this miRNA which regulate expression of EPST1 gene. So these two genes are very important for development of disease development of Alzheimer's disease. Hence, it is a significant and promising biomarker for disease detection of SCA and AMDI, which is early diagnosis of Alzheimer's disease as compared to healthy individuals. That's all. Thank you. These are the references.
Thank you. Thank you. How to say your name? Yi-Eun. Yi-Eun, is it?
Yes.
Yi-Eun. Yeah. Thank you so much for your presentation. Okay, I want to hear one question from the student. Anyone, ask question. You can't be just quiet like this because we are having tutorial. It should supposed to be a discussion. So ask question.
I would like to know from you what is the disadvantages of this essay and which
Yeah, did you presented?
Microarray
Yeah, okay.
advantage and disadvantage. Okay,
Yeah.
so it relies on hybridization, in which the hybridization means this.
Let's go.
So you have your circular RNA that is your sample, and then you have to hybridize on this DNA chip. However, this hybridization method will somewhat not so specific as compared to direct alignment of RNA sequencing. So this is one of the limitations of microRNA. Then the next limitation is that it is only available to analyze the predefined sequence, also because there is the presence of this probe. So you have to know what is the sequence first to make the probe using the arrayer system. Yeah, this is the disadvantages of microarray. Is there any question?
Yeah, thank you so much, Ian. It's a very good presentation, compact
you.
and precise. Okay. So thank you so much. Okay, for the next presenter, who wants to go next?
I'll go next.
Dr.
Yeah. Okay.
Yeah, you can go, Leo. Leo or Yao?
Yao.
Okay, come.
Hi, Dr.
Share your slide.
Daru. Okay. Okay. Okay, I start my presentations. Okay, very good evening to the Dr. Daru and my fellow friends. Today, the topic I'm going to present is the RNA sequencing for biomarker discover, workflow, and the considerations. Okay, before we dive into the workflow advantages and limitation of this approach, let's see a quick background on this topic. Biomarkers serve as indicators of the physiological or the pathological process. So it's very important in the disease diagnosis, prognosis, and the treatment monitoring. There are various biomarker type assists, for example, the genetic, proteins, and the RNA-based biomarker. Each of them offering a very unique insight into the disease mechanism. Transcriptomics involve studying RNA molecule within a cell or tissue, providing comprehensive insight into the gene expression and regulations. Transcriptome-based biomarker like microRNA offer a potential as a disease marker due to their involvements in the post-transcriptional gene regulation. Transcriptomic analysis such as RNA sequencing, RNA-seq, enable the comprehensive profiling of RNA molecules is very helpful in the biomarker discovery and the validations. Okay, what is the RNA-seq? RNA-seq is a sort of RNA sequencing. It's an advanced method employing the next generation sequencing (NGS) to assess the quantity and the sequence of the RNA molecules within a biological sample at any given moment. RNA sequencing also revolutionizing the study of the transcriptome, as it is highly sensitive and accurate tool for the gene expression study and the marker discovery. Compared to the previous Sanger sequencing and the microarray based technique, RNA sequencing offering better coverage and the resolutions of the transcriptome's dynamic nature. Additionally, not only quantify gene expression, RNA-seq also enable to discovery of a new transcript, detections of alternative splice genes, and also identifications of allele specific expressions. So the RNA workflow initiate with the extractions of the RNA from the biological sample, such as a tissue or cells. So following this step, isolations of mRNA or specific RNA from a total RNA is achieved through the method like poly-A selection, ribo depletions, and size selections. Subsequently, the RNA is converted to the cDNA through a reverse transcription process before entering the library preparation. Before reverse transcriptions and the library preparation, it is important to assess the RNA quality if the RNA is degraded. If yes, the RNA is degraded, then the specimen treatment can be actually applied to repair the degraded mRNA. If it's no, then the workflow can be proceeded with just now as mentioned, poly-A selection, ribo depletion selection, and size selections. So each step in this process is very crucial for obtaining the high quality sequencing data. During the library preparations, due to the various length of the different mRNA transcript, so fragmentations of a complementary DNA is important to ensure the uniform size of the sample. So this step is very important to enable the consistent sequencing. Okay, sorry. Then following the fragmentations, there is a sequencing editor are ligated to the complementary DNA fragments, allowing them to bind to the sequencer itself. So this process is also called a ligations process. Subsequently, the prepared sample are placed into the sequencer. So after the library preparations and we put the sample into the sequencers, you will undergo a step called cluster generations or the clonal amplifications. A DNA strand will bind to the adapter on the flow cell of the sequencers. Then the strand and then bend over and then bind to the other adapter on the flow cell. So through the bridge amplifications, each library fragments act as a seed to generate a clonal cluster containing thousands of identical fragments. And after the bridge PCR or cluster generations, it's important to assess the concentrations of the prepared library to prevent the over clustering or under clustering. Because the over clustering, it will cause the sequencing run fail because this is high density. Then for the under clustering, it may result in the reduced sequence output. Okay, so after the library preparation, sequencing by synthesis follow. The templates with a primer for the synthesis bind to the flow cells. Then complementary nucleotide with a fluorescent dye will bind to the templates. As you can see, A nucleotide is bound to the sequence. Those unbound will be washed away. Then after this binding occur, okay, there will be a fluorescent emissions and response to the light stimulates from the nucleotides. So then this image will be captured by the sequencer. So following this binding and imaging, the fluorescent site is cleaved from the nucleotide and rinse off. So this process will repeat and until the full sequencing have been read. So has been complete. So the resulting sequencer read will later undergo the data analysis. For RNA sequencing data analysis, it involves comparing the resulting sequence read with a reference database. So this process aims to align and map the sequence read obtained from the RNA sequence experiment to a known reference genome or transcriptome. Okay. After RNA sequencing analysis, qPCR serve as a critical method for the validating the RNA sequence result due to its high correlations in relative gene expression result. It offers an independent validations of the sequencing accuracy, confirming the biological interpretations derived from the RNA sequencing experiment. So this can ensure the trustworthy and the reliable data for the publication purpose. Next, let's dive into the advantages offered by this approach, highlighted as follow. First is a high resolutions, massive high throughput, high sensitivity, and ease of use. So RNA-seq can accurately distinguish the individual base. Therefore, problems such as the background noise and the cross reaction caused by the fluorescence analog signal can be effectively avoided. And for through the transcriptome sequencing technology, hundreds of millions of base sequence can be obtained, which can be basically cover the whole transcriptomes. So it can offer a very comprehensive insight into the gene expressions and the regulations. The rare transcript as low as a few copy in target cell can be detected. Due to its high sensitivity, it can enhance the ability to capture and analyze a low abundance transcript. And this technology can be used to analyze the whole transcriptome of various species and does not need reference genome or design specific probe before sequencing. So it's very easy for use, very easy to use. Okay, certainly the approach has its disadvantages, including the potential limitations, such as RNA degradations and sample preparations.
You may summarize your presentation.
Okay, so actually I'm going to be end of my presentations. It's just that these are the disadvantages of the RNA sequencing, RNA degradations. As we know that RNA is very fragile, so it's in the instability of the RNA, so that's why it's tend to have a degradations. So if samples are not immediately processed or stored properly, then the RNA degradation can affect the quality of RNA extract. Then and also for the RNA sequencing, you need a lot of computing power. So because we need a lot of advanced informatic tools and the computation skill to process and interpret the data effectively, and also lack of the standardization in the RNA sequencing across the different lab and study also will impact the accuracy and reliability of data. And also finally the higher cost. Higher cost is one of the disadvantages of the RNA sequencing. Okay, thank you. Yeah. Hello.
Okay, thank you so much, Yo, for the presentation. May I have a question from the floor?
Doctor, can I have a question?
Yes, please.
Is it I do the presentation in the wrong direction?
No, it's okay. No worry.
Okay.
It's okay. Don't worry. And after all, this is not your assignment. This is the tutorial. The assignment will be your written report. So please do the written report properly. Okay.
So the written reports that's supposed to be based on one article, then I have to discuss the
Methodology. Methodology for the discovery. Yeah. You see how Yen presented. That's how you have to do.
Okay, understand.
Okay. So any question from the floor? Kavita, please ask one question.
Yeah, I have a question.
Yep.
So do you think that RNA sequencing is the better method into studying this rather than using microarray or nanostring technology?
If compared to the nanostring technologies and the microarray, I think RNA sequence is the best method to sequence, as it can be sequenced high base pair, long base pair, long read. It can be sequenced long read.
Okay,
Mm.
thank you.
Yeah. So when you can sequence, I mean, wide range, but end of the day, when it comes to clinical samples, you have to focus and be very specific. But this platform is not that specific, right?
Mm, Actually, it depends. For example, like just now that we can say that we can depends on before the convert the RNA into the DNA. Actually, there's a selection, right? So we can use these poly A selections or ribodivisions to help us to select the desired transcribe. If you want the transcribe, I mean, which is a shorter one, or if you have poly A attached one, then we can use this way to help us.
Okay, thank you.
Thank you. Yes. Any question more?
Okay, thank you so much.
Thank you, Doctor.
No, I'm you. Can you hear me? Okay. My laptop hang. Okay, let's go to the next presenter. Who wants to present?
Oh yeah, I will go next.
Yeah, please share your slide.
Okay. Okay, so hi, good evening to my tutor and also my friend. So today I'm going to present about transcriptomic biomarker discovery, microRNA profiling with nanostring technology, which is a method given to me. So my name is Leo. So yes. So okay, so when you're talking about neurodegenerative disease such as Alzheimer's disease or schizophrenia, right? The usual the method to scan for the biomarkers is to scan through from the cerebral spinal fluid (CSF). However, in this kind of disease, in this kind of sample collection, this way of sample collection requires very skillful physicians lah. So yeah, CSF biomarkers require invasive and delicate lumbar procedure. So thus in this discussion, I'm going to discuss the alternative source, blood, which consists of the microRNA as the potential biomarker that can serve for diagnostic and prognostics and treatment progression purpose for Alzheimer's disease. Okay, so what is Alzheimer's disease? So in a very short way, Alzheimer's disease is a progressive neurodegenerative disease that affects millions of people worldwide, especially, of course, the elders. It's the most common form of dementia mentioned by Ian just now, causing a decline in memory, thinking, and behavior. All right, so this is the, So this is the global distribution map of Alzheimer's disease. The D represent for death and P stand for the patient. So as you can see that in Malaysia, in Southeast Asian region, right? So there's approximately total amount of 10 million plus of patients and causing about 500,000 deaths. All right. So why we choose microRNA? MicroRNA belongs to the class of non-coding regulatory RNA molecules of 22 nucleotide length that modify gene expression at the post-transcriptional level. So in many studies, microRNA has found to be linked with Alzheimer's disease. Example of these microRNAs, HSA-MIR106 has been, and its brands have been demonstrated to target the beta amyloid precursor protein, which contribute to the amyloid plaque, which is the main factors of causing the Alzheimer's disease. And yes, and then these other two, there's that demonstrate to target the BACE1 beta secretase, which splice this beta amyloid precursor protein into the proper protein, a functional protein. So other than that, microRNA has found to be playing role in disease etiology as well. So microRNA profiling is necessary to determine all related microRNA AD. So this this approach allowed researchers to profile global microRNA expression level using a relatively small amount of purified RNA in a specific and sensitive manner. Okay, so generally this is the working flow of the experiment design. So we have two cohort core 1, which consists of 11 Alzheimer patients, 9 mild cognitive impairment, and also 20 healthy patients. And then core 2 consists of 20 Alzheimer's disease and 17 age-matched non-healthy samples. So once you have this person, we get the blood serum from them. So we'll do a total RNA extraction using this from the article itself. It used this Microvana Paris kit from Knife Technologies. Then after that, this sample was then used for this high throughput expression using the N-counter microRNA expression assay by NanoString in triplicate. And last but not least, the validation is done by using qPCR. Okay. So this is a flowchart of NanoString technology working pipeline. So as you can see, we have samples. So from the samples, we will do the DNA extraction. Of course, in this case, you have RNA. The starting sample is microRNA. You have to convert into a cDNA first. So after that, you will do a hybridization. This is important for the next step, because next step is we'll do purification, alignment, and embolization. And the machine itself will do analyzer from us, okay, doing imaging and counting. So this is the result of the microRNA they found to be related. This is a plasma microRNA. The microRNA exists in your blood with the fold change and the p-value. So as you can refer to here. And last but not least is the pros and cons of using NanoString. NanoString has said to be specific and sensitive, as it's a direct digital counting and distinguish closely related microRNAs, and then is versatile and multiplexing. Of course, it's a large panel. It can use for large throughput, and you can customize the NanoString, the analyzer itself, and ease of use and speed, simple workflow and faster result. However, the disadvantage is the slightly higher RNA quantity requirement for the NanoString assay. Sometimes, if you do these RNA samples, dealing with RNA samples, the quantity might not be as much as you like, even though you have performed the protocol well. So approximately 100 nanogram is a minimum. And inability to detect unexpected novel fusion gene variants. And last is a costly technology, like if your throughput is not high enough. All right, so basically that's it from me.
Okay, Leo, I have a question. How do you mean by costly technology if throughput is not high enough? What does it mean?
Okay, so what I mean is that this technology, right, the machine itself for you to get is very expensive. Of course, if you compare to RNA sequence, right, if you're doing a large scale samples, like large sample size, is more cost effective. However, if you are just using a very starting with small sample size, right, then the cost, it might not as cost effective as it should be.
Okay, then any question from the floor?
Leo, I want to clarify something. Just now,
Yeah.
during your method, you mentioned that the RNA was converted to cDNA, right?
Yeah.
But when I thought this technology, NanoString technology, we don't have to convert, we can directly use the transcript. Am I wrong?
Oh yeah, sorry. Yeah, I was confused by this chart. Yeah, true. Yeah, you're correct.
Okay, thank you.
You have to correct this figure again. Okay. Anything?
Can I ask a question?
Yes.
What is the customizability? You mentioned its advantages. You said that customizability, if you do something with the machine, something like that.
Oh no, this is mainly due to for the multiplexing purpose. Like actually, you can do a lot of different customizations to with the parameters and the starting sample setup like that.
All right.
Okay, then thank you, Leo, for a very good presentation.
Okay, thank you.
May I know how many more presenter left? Okay, please, next presenter.
My topic is RNA sequencing reveals for macros. RNA sequencing is a high-throughput sequencing technology used to measure and analyze the molecular weight and relative quality of all RNA in cells or tissues. Through transcriptional research, RNA sequencing reveals information on gene expression, splicing variations, and the discovery of new genes. In terms of biomarker discovery, RNA sequencing identifies potential biomarkers that may affect the diagnosis, prognosis, and treatment by comparing gene expression differences under different conditions. The basic process of using RNA sequencing to discover biomarkers is as follows. Firstly, extract the total RNA from the sample. Afterwards, the establishment of an RNA library will be carried out, which includes reverse transcription of RNA into cDNA and the addition of adaptive sequencing marker for high-throughput sequencing. mRNA enrichment can be chosen to focus on the information of transcripts. During the library construction process, a series of quality control steps are implemented, including total RNA quality detection of cDNA synthesis efficiency, selection of library fragment size, and the evaluation of library concentration. This helps to ensure good library quality and provide reliable data for subsequent sequencing. Afterwards, sequencing and data analysis will be conducted. Gene expression analysis, differential expression analysis, and the functional enrichment analysis will be performed on the sequencing data. Gene expression analysis can reveal the level of gene expression in the sample, while differential expression analysis is used to compare changes in gene expression under different conditions. Functional enrichment analysis helps to understand the biological functions of differently expressed genes. Based on the results of differential expression analysis, screen for possible biomarkers. Gene will significantly differential expression can be selected to further validate their importance in disease or other conditions. Further experimental verification will be conducted to confirm the reliability of biomarkers. Among them, qPCR is a commonly used method that can quantify the expression level of specific genes and verify their differential expression in RNA sequencing results. RNA sequencing has the following advantages. It can detect all transcripts, including new transcripts and non-coding RNA. It also has high sensitivity to low expression genes and small amounts of RNA molecules. Having high resolution can provide information to the on expression level of individual transcripts and accuracy determine the partners and level of gene expression. And the dynamic range, capable of detecting wide dynamic range of gene expression, including genes with low and high expression levels. And it also can detect unknown genes and transcriptions. High reproducibility to ensure the reliability of results. And it has a wide range of application in various fields such as disease diagnosis and drug development. Although RNA sequencing has many advantages, there are also some drawbacks. Complex data processing. RNA sequencing generates a large amount of data requiring complex data processing and analysis, including quality control, comparison, differential expression analysis, and other steps, and the high cost. And the reliability and the accuracy of the results are limited by the number and the quality of samples. So it is necessary to strictly control the sample collection and the processing process. Okay, that's all.
Thank you so much, Jai. Is there any question from the floor?
That's one flow chart.
Anyone wants to ask question? What about Sana? Sana, can you hear me?
Yes, teacher.
Sana? Yeah,
Yes.
can you ask question?
About Song's presentation. Sorry, like I don't have any question about but I cannot see validation method first. So can you just tell me how you validated your method?
What's the question? How validate the macro?
Yeah, you use it to RNA sequencing, isn't it?
This article is used qPCR to.
Oh, okay. I got it because I couldn't like find out in your slides and your presentation.
Okay, is there any more question? No? Thank you, Jai, for the presentation. Now we move on to the next presenter.
Teacher, can I go for the first one? Next one, sorry.
Sana, you want to present?
Yes, I want to, yeah, please. Can you see the presentation? Sorry.
Sana, can you present? Start presentation.
Yes, I want to, but I shared the slide. Can you see the slide? Sorry, better. I don't know how to share it. Like, I think uh,
Sana?
yes, sir. Like, I want to share it, but I don't know how to share my slide.
Okay, never mind. You try to fix your error. Next presenter. Someone else present first. You try to check the technical part, Sana.
Okay.
Okay, we move on to the next presenter. Then after the next presenter present, then you can come in again, okay? Who's the next presenter? Is this your slide? Yeah, okay. There you go. You have a slide. Okay, you can start presenting.
Okay. Sorry. My presentation that being chosen by doctors, like it's about nanostring technology. I really like. I want to clarify that and like maybe I being misunderstood about how to prepare my presentation or how to deal with this subject. So I will just cover up the nanostring technology about my topic because I wasted all my time to finding the papers because I thought like all my presentation is gonna be about my paper. So today I will present about nanostring technology, predictive value of circulating microRNA in lymph node and metastasis for colon cancer.
Hello, Sana.
Yes, but I don't know, like my slide is not working.
Your system is hang, is it?
Yeah, it's fine, but I don't know, my slide is not going on. So like, I think if there is anyone go, it's gonna be much better. Okay. So, my content is gonna be background, objective, outcome, and flowchart about mythology of this method. So, my background is gonna be about colorectal cancer and finding biomarkers, microRNA that related to the recurrency of this lymph node metastasis of this type of the cancer, and they at the gap of in the literature, they found that there is no any predictive biomarker for completing TNM classification step in the cancer management system in the colorectal cancer. So my objective, sorry, I don't want to go through everything, but only this method, like in this study, I will use, they use it nanostring technology for gene expression level profile. What's nanostring technology? Nanostring technology, it's a nanostring encounter with a multiplex nucleic acid hybridization technology that allows for the assessment of gene expression or fusion transcript in high throughput manner. The technology requires only a little amount of the sample. It's 1 nanogram of RNA input per sample without any amplification, because this method is not about the enzymatic amplification of any genes, only detecting and identifying the gene being expressed in the cell, in the sample. So here we can see the molecular barcode. We can see the molecular barcode idea. It came from the two parts of the complex, like target specific reporter probe and also target specific capture probe. The barcode one, it's work on the identifying the specific gene of the RNA, but the target specific capture probe, it's bound with the biotin that help with the imaging procedure steps in the nanostring technology. Sorry. Nanostring technology steps, first, like we have capture probe and reporter probe. After they will capture with the specific microRNA or RNA in the sample, then they will form the complex target probe complex. After formation of the target probe complex, then the solution phase hybridization happen, and then all the sample in single tubes will be washed from the excess probes and excess probes inside the sample. After the washing phase, then there is a immobilization phase. Immobilization phase happens with the help of the biotin in the capture probe. After immobilization, and then the target probe complex will be bound to the surface, and from the surface of that technology, then it will be created by the software system. The software about the barcode. Each barcode has different colors, and the different colors is designed for the different genes, as they mentioned, like my friends. Nanostring technology mostly like will count or identify 800 of gene expression micro RNA in the sample. So what are the advantages of the nanostring? First of all, like it's flexible sample requirement, no need for a lot of amount of the sample, and only for the pipetting, there is 4 steps for pipetting, so avoid any contamination, anything related to the sample. No amplification, there is no amplification enzymatic reaction, and also like as they mentioned, like 8,000 hybridization in single tube will happen. and its sensitivity and precise of the capturing the RNA in the sample and quantitative. It's real quantitative in the explicit level of the RNA in the cell, in the sample, and simple and complement inside one's system. Okay. Disadvantage of this sample in this technique, one limitation is the need for normalization of expression level with a return gene which can introduce variability in the result. It's a proprietary platform that offers a limited flexibility. It's customizable, we cannot deal with or change to our purpose. Nanostring doesn't provide a single cell resolution for single cell mutation or any change in the RNA transcriptomic. Spatial information at the single level of the cell. This is outcome of my sample. First, you can see the methodology flowchart of the study that I studied about the papers. Sorry, I just misunderstand everything. First, in my methodology flowchart, like sample collection, they collected 79 blood samples of patients, and then the sample coverage two sets of their cohort, discovery set and validation set. In the discovery set, 24 of the sample being undergone the nanostrength assay for discovering the microRNA within the sample, and the validation set, they did the validation step for 79 of the sample after they found the microRNA biomarker in the 24 of the discovery set. So the validation done by the quantitative real-time PCR, a Chapman process assay. So then they found about the outcome about this validation, the discovery. They found that there is biomarker related to lymph node positive group that being upregulated or downregulated, and also the validation. In the validation step, they found it like out of the 10 of the biomarker microRNA, there is like 3 of them is significant with the lymph node positive metastasis patient. First, you can see analysis of 24 plasma samples from colon cancer patients using quantitative real-time PCR after validating the result from the NanoString technology. And here's the 3 microRNA. Firstly, they validated the three of them in 79 sample, and then they validated the 3 of them that related to the lymph node metastasis. Sorry. Here's my response, and the discussion is about the paper. Sorry, NanoString offers. They found it like there is a hope for provide this technology provided to the help of the finding predictive biomarker in colon cancer for the patient with LN metastasis. NanoString technology provided a powerful tool for studying gene expression biomarker finding in cancer. In this study, for example, NanoString offers several advantages, including the ability to analyze multiple biomarkers simultaneously, and sensitivity, reproducibility, technical robustness, and utility for clinical application. Physics ability, and this is my reference. It's not all included, but this is most of the reference that I prepared my presentation. Thank you so much. Thank you. Any question?
Okay, Sana, thank you for a very good presentation. I think you have done very well. You have presented on the discovery and the validation, so it's quite good. Well done. So any question from the floor? Ian, can you ask one question?
All right. Sana, can you go back to the method? Not the method, the flow chart of the the nanostring technology, like the first few slides.
Okay, this step,
Yeah, this one. So is there no amplification of the sequence?
Yeah, this is one of the characteristics of nanoshrink. Apart from
Okay.
sequencing and quality piece outing, it's not
So
any amplification. There is no any enzymatic reaction going on inside the sample in the technology.
if there is no amplification, is there a limit of detection?
Yes.
Means if it is very less, then you cannot detect with nanostring technology.
This is one of the drawback of this technology, but they develop it in that way that it can specifically detect the lowest amount of the RNA in the sample. So in that way, like it's balanced the way that it works.
Okay, all right, thank
And
you.
this is one of the advantages of this technology. Like the sample, when we are collecting the sample, as like Kavita and Leo, you mentioned like frozen from formalin, frozen sample, it won't be affected by the frozen and the treatment of the sample because it will, there's only like detecting the RNA. It's not about the RNA being degraded, like anything happened to the RNA in the sample.
Okay, thank you.
Thank you so much for your question.
Thank you, Sana, for the presentation. Now we move on to the next presenter.
Okay, Dr. Me.
Yes, please share your slide.
Okay, now. Okay. Victor, hello.
Yes, please share your slide. Okay, I can see
Okay.
your slide. Please start your presentation.
Biomarker discovery, transcriptomy, breast cancer as case study. Name Rad Habib. Introduction. Biomarker objective including of the person medical state myostable according the reproduct from outside patient according the WHO biomarker are structure substance present in the body they can measurement influence a product outcome disease streptocomic technological such as RNA sequence and microarray using a transcriptomy analysis. The appropriate complement a gene expression analyzes the genetic expression level is one of the more useful methods understanding requirement change natural of the concert cell. This model organism target molecules. Transcriptomy technological stand tools. The analysis transcriptomy biomarker. Cancer biomarker identified. The example, gene expression different between normal and cancer tissue. Define gene, auto expression under expression cancer related patient biomarker. A specific RNA molecules at indicated abnormal gene, decade use molecule at early cancer diagnosis target therapy. Provide the molecule under the cancer development. The challenges and solution, the cancer discovery using a transcriptomy. Challenges: efficient tissue analysis, vast, complex transcriptomy data solution. Advantage: bioinformatic tools. Use bioinformatic tools, the data analysis. Okay, the case study of HER2 gene, the breast cancer example, the complex, the breast cancer invite multiply technology, the gene expression analysis, auto gene expression level, a breast cancer come of the normal cell, automized technology.
Right.
Yes, doctor.
Can you hear me? Okay. Okay, first of all, what is your discovery method that you use for the, what is the discovery method that you use?
Doctor, just a minute. They use the RNA sequence.
RNA sequencing, okay.
Yeah.
So for the discovery of...
Yeah. Automized technique such as polymerase chain reaction.
For the validation, what is the method you use?
Doctor.
Yes. What is the method for the validation that you use?
Doctor, advantage and disadvantage. The use the RNA sequence analysis of the gene expression, the breast cancer. Okay.
Okay, can you go back to your slide? Go back to your first slide.
Okay, doctor.
Yeah, no, go back to your first slide. I just want to see your slide. Can you please go to your first slide?
The slide?
Yes. First slide. How do I call your name? Ra'at, is it? Is it Ra'at?
Yeah.
Okay, go to the first slide, Ra'at.
First slide?
First slide. First slide. Okay, go up. Yeah.
Yeah.
Okay. Go to the second slide.
This.
Okay, next.
Yeah,
what is which article you use as a reference? Have you picked up any article?
Yeah.
What is the disease that they study in this article?
The disease, breast cancer disease.
Breast cancer disease.
Yeah.
Okay.
The data analysis by the RNA sequence.
Okay. How many sample size they use?
10 sample. Between 10 sample normal, abnormal 10 sample. The change between the patient, change between the patient, can you detect between the patient?
Okay.
By using the bioinformatic tools.
Okay. Move on to the next person. Means that you have an article you use as a reference, because your presentation earlier looks a bit general. Okay.
Okay.
Move on to the presentation. Carry on with your presentation.
Okay.
Yeah, carry on with the presentation.
That's all.
Present.
Okay, doctor. This is all a presentation.
Okay. Have you finished presenting?
Okay, but found advantage and disadvantage RNA sequence.
Okay.
Yeah. High percent gene expression analysis. See capability of the analysis, a small sample, and disadvantage, high cost, analysis large data, requirement, advantage source. Application of the transcriptomy using the translation of the transcriptomy data, micro early diagnosis based of the progressive impact, development of the treatment therapy. This discussion. This complex of the study breast cancer case, transcriptomy analysis relays provide the gene expression patient ability early detect reference polarized treatment stage of the on-gene address understanding of the problet certificate of the transcriptomy research, cancer diagnosis and treatment. In the Kerry transcriptomy study remained fundamental objective twin toward the percent Kerry relation of the cancer, result of the cancer. Okay, thank you, Doctor.
Okay, thank you, Rahat, for the good presentation. Any question from the floor? Any question? No question? Leo, can you ask one question? Leo, can you hear me? Is Leo there? I hope all of you can stay until the end of presentation, okay? Don't leave the presentation until the presentation complete.
Yeah.
Okay, Kavita, can you ask one question since you said yeah?
Okay, you said you did, I mean, the study did RNA sequencing to get the HER2 in the breast cancer, but any other platform was used to validate this?
What, Kavita?
In your study, RNA
Yeah.
sequencing was done
Yeah.
to find, and they discovered that HER2 was the biological marker, right?
Yeah.
But was there a validation with other platforms?
Yeah, change between the patient.
I mean, was there a validation using other platform like microarray or nanostring?
RNA sequence.
Okay, Ra'an, what is the question is, you do a discovery using a RNA sequencing. So did you do any validation using other platform? You know, like in your write-up, if you don't do that now, in your write-up, in your assignment write-up, you have to propose this validation method for your study. So in case that you use RNA sequencing to discover marker in breast cancer, you must have a validation using other method. If it's not being proposed in the article that you read, you may suggest that yourself. You can use like RNA sequencing for the validation method, that's fine. Do you understand, Ra'an, what I'm saying?
Yeah.
Okay, so if you don't include that in your presentation, please include that in your write-up.
Okay, Doctor.
Okay? Okay, thank you. So may I know
you.
who is the next, who is left with the presentation, who is not presenting yet? Okay, we have two more, I think. Is it two more? Puspa and also one more is who? Sultan B, is it? Yeah. Okay. So can you do a presentation now since 2%? So I will not break for prayers. I think we just complete the presentation, then we will just off the presentation. You can go and pray, okay?
Okay, doctor.
Okay, next presenter, please.
Is my slide visible? Is my slide visible?
Yes, I can see your slide. Please present.
Okay. Okay. My topic is RNA sequencing identifies novel transcriptomic signatures in intestinal failure associated liver disease. Introduction. Intestinal failure associated liver disease, IFALD, refers to liver dysfunction caused by long-term dependence of parenteral nutrition, PN, in patients with intestinal failure. So the clinical spectrum of IFALD mainly includes steatosis, cholestasis, and gallbladder sludges and stones. To date, the mechanism underlying IFALD development are poorly understood, and there is no effective therapy other than discontinuing the perinatal nutrition. So methods, the animals they used were male Sprague Dowley rats, age of 4 weeks, subjected to 0.9% saline with standard chow, control group, 12 controls, and TPN for 7 days with 16, and TPN for 14 days for 4 samples. So the TPN is the total parenteral nutrition solution, has an all-in-one balanced mixture of 8.5% amino acids, lipids, and 50% dextrose, electrolytes, trace elements, and vitamins with a non-protein calorie to nitrogen ratio of 295 is to 1. So coming to the RNA isolation and RNA sequencing, RNA extraction by triazole reagent extraction. Next, library construction and sequencing done by Illumina High Sequencer 4000. Next, sequence reads, trimming, and alignment to rat genome RNOR 6.0 by using TrueFat2 software. Next, gene expression estimation using cufflinks software. Differential expression analysis, DEGs, using DE sequence 2 package with a threshold of fold change more than or equal to 1.5 and FTR less than 0.05, where set as threshold. Gene ontology annotations by using BLAST2Go, COG, clusters of orthologous groups of proteins, database alignment by predict and to predict and classify functions, Keck pathways assignment by using Perl script, gene set enrichment analysis, GSEA, cluster profiler R package, active transcriptional factor network by using CoRegNet bioconductor package. So the validation in the study was done by real-time qPCR. cDNA was generated from total liver RNA and the real-time qPCR was performed. Validation, the real-time qPCR analysis showed that HES6 expression in the liver revealed a consistent decrease in its levels following 7 days of total perinatal nutrition infusion compared to the control group. Furthermore, there was a tendency for further reduction in expression at day 14, as illustrated in the figure. Conclusion, HES6 was identified as a potential regulator that transcriptomic factor activity analysis unveiled significant interactions between HES6 and multiple activated transcriptomic factors in IFALD, with HES6 and NR1D1 exhibiting the highest interaction. The RNA sequencing analysis and subsequent validation showed a decreased expression of hepatic HES6 in TPN-fed rats, suggesting potential link to IFALD development or progression. In summary, findings shed light on the intricate molecular landscape of IFALD, identifying HES6 as a potential regulatory factor. Advantages of RNA sequencing is quantitative measurement, detection of novel transcripts, high sensitivity, global transcriptome profiling, not requiring extensive technical knowledge, and dynamic range. Disadvantages are the cost, computational complexity, RNA degradation, sample preparation bias, biases in library preparation, limited coverage of low abundance transcripts. This is a reference. Thank you.
Okay, thank you so much. How do I call you, Sultana?
Yeah.
Okay. So can you go to the disadvantages slide?
Yeah.
So how do you mean by sample preparation bias? What does it mean?
It would mean that the authors would like to select only a specific gene, like a specific RNA expression.
Are you sure? Anyone? Anyone wants to add? Who's doing on RNA sequencing? Anyone wants to add? What does it mean by sample preparation bias?
The sample preparation covers amplification part or converting the RNA to cDNA.
Sorry, I cannot hear you. Come again?
Oh, no.
Does the sample preparation involves the conversion to RNA to cDNA?
No.
Yes, Sultana, please answer.
No, they directly sequence it to Illumina and then generate the library.
So the preparation is not any from RNA to cDNA, or could there be any amplification method involved?
No.
Then I'm not sure why there's a bias.
Yeah. So okay. My concern is whatever, not for this presentation, for any presentation that you do, you're gonna take research project later on that you will have a lot of presentation. You're gonna have proposal presentation, research presentation, final presentation, and viva. Please make sure whatever that you put on your slide, you have to understand everything that you put on the slide. You cannot just put on the slide and when somebody asks you what does it mean, that you cannot explain. You got what I mean? You must know exactly what is there on the slide. Okay? Not for this presentation, for any presentation, and not only for Sultana, this is for everybody. Okay? Okay, then Sultana, thank you for the presentation. We move on to the last presenter. This is the last presenter, right?
Thank you, Doctor.
Pushpa, you're going to be the last presenter, right?
Doctor, we still have Zhe Yu, Gao Zhe Yu.
Yeah, where you went? You went to washroom?
Me?
I cannot hear you.
Yeah, just now I typed already, I went to washroom.
I called your name just now. I couldn't hear you. No, it's okay, no problem. So now we are on the last presenter. Pushpa, because we have a Maghrib prayers. So we finish the last occasion. So we don't have to have a break, okay? We finish the presentation. And then everybody can just take our desk and then that Muslim can go for prayer. Pushpa, please share your slide. Okay, you may begin the presentation now. Hello, Pushpa, can you hear me? I cannot hear you. Did you unmute your mic?
Hello.
Can anybody hear Pushpa?
Hello.
Yeah.
Yes, Pushpa. You can continue, yes. Can you see my slide? Hello.
Yeah, okay. Carry on.
Hello, everyone. Today's title for my presentation is Identification of Plasma Biomarkers of Human Intracerebral Hemorrhage Subtypes through Microarray Technology. Introduction. Intracerebral hemorrhage. It is a condition in which the blood vessels rupture in our brain. It accounts for about 10 to 15% of all strokes, and it's associated with high morbidity and mortality rates. It is caused by either hypertension or cerebral amyloid angiopathy, and so far there has been no any effective treatment for it. The objective of the article which I referenced to was to identify potential biomarkers that could differentiate between CAA and HTN ICS based on like gene expressions. Methodology. The process began with the collection of blood sample and then extraction of RNA and then creating a complementary DNA library. Then from cDNA, cRNA library was prepared, and using this library, gene profiling was done in using GeneChip Human Exo 1.0 stat array. Then the datas were pre-processed using RMA method for background correction, normalization, and summarization of the values. Then differential gene expression analysis were conducted, and only the transcripts that showed differential expression were selected, and then they were subjected to hierarchical clustering to identify common patterns of expression. And then the data were again analyzed using R language and bioconductor libraries. Then a replicative study was conducted using, and then here they used quantitative real-time PCR for validation. The samples were collected from 18 patients, and then they extracted RNA and then did the PCR and then analyzed using Biosystems SDS7900 system software. So unfortunately, unlike in the microassay, while in the validation process, they could not confirm all those genes that were generated in the microarray. So only one gene were confirmed in the validation process. The result. Advantages and disadvantages. Advantages, microarray is like it has high throughput, it is cost effective, it is cheaper compared to other assays, it is time efficient, it is quick, easy to perform, and then it helps to like generate hypotheses about the underlying molecular mechanisms involved in biological processes or diseases. Disadvantages is that there might be cross-hybridization. It requires prior knowledge of the genes that were being analyzed, and then also like it might not its like level of expression is low compared to other assays.
Okay, thank you, Puspa, for the good presentation. Yo, since you came back, can you ask one question? Yeah.
Yes. Okay. So just now again, you are doing the microarray, if I'm not mistaken.
Yeah, minus microarray.
Yeah. Again, my question is, why do you think microarray is they want to use microarray in this experiment rather than RNA sequencing or nanostring?
To be honest, this paper dates back to like it's a paper of 2016. So back then RNA sequencing wasn't like available like microarray. So like at the time when they did this research, RNA sequencing were just there on the market, but obviously they cannot afford it because it's obviously expensive.
Okay, so now because at 2023 the technology is available already, do you think that changing the method of study will lead to a better outcome?
Oh yeah, obviously, yes, absolutely.
Okay.
Thank you.
Thank you.
Okay, thank you, Pushpa, for a presentation. Thank you, Leon, for your question. I think we have to end the presentation today. Thank you, everyone, for contributing for today's presentation tutorial. I hope that you can do a good write-up. Please take note of all the comments that I have given today focus only on the discovery. Do not write anything about the result from the article. I do not want to see a diagram, bar graph, pie chart, all that I don't want to see. That's not important for your assignment. Focus only on the discovery method. How do you discover a biomarker using a tool that was given to you? And please focus on the validation part. For those who have missed the validation in this presentation, please do that in your assignment. I have already written the criteria of the assignment in the e-learn. Please have a look on it. Please make sure you don't do plagiarism. The plagiarism must be below 20% for your assignment. And please upload. When was the date that I agreed to give you all the other day?
It's 12th of
12.
January.
Okay. So please upload by 12 January into the e-learn, okay? Please.
Doctor, I have a question. Like, for example, I thought that we have to choose a disease and biomarker discovery by the, the method that you choose it for us. But in the presentation, I saw like it's not about the study that we choose. It's only about the discovery method, like
Sorry, Sana, you talk too fast. Sorry, sir, you talk too fast. I cannot catch what you're saying. What is your question? Come
Sorry.
again. Talk slow.
My question is that I got it like that. My assignment is gonna be all about one papers that I chose a disease. Like for example, I choose colon cancer, and then I have to talk about the method that they use it, the one that you choose it for me, nanostring technology. And then finding the validation method for the finding they found it by nanostring technology.
Yeah.
So then I have to mention the advantage and disadvantage of nanostring methodology, and that's it. Like I thought
No,
it's
you
gonna be
also
like
have
that.
to cover on the validation. Okay, if let's say your discovery method that I have given you is nanostring, so
Yes.
now you have to use a validation method. Just take from the paper any validation method that was given, or you may suggest the validation, like maybe RNA sequencing, validate through RNA sequencing. So now talk.
Sorry, doctor, like in that case, I have to write about nanostring and the validation method, for example, quantitative PCR. Explain everything about
you're
them.
right.
Okay.
Yes, and then the advantage, and then please cover on the reference, and please remember, put it a flow chart, okay? It's like the discovery method. If let's say you are doing on nanostring, so what is the step-by-step that you have to do in the nanostring to discover your biomarker? Put it in the flow chart.
Okay.
Is everyone clear with the assignment that I've given? Okay. So if everyone clear, I think that's all for today's today's class. So thank you,
Sorry,
everyone.
doctor.
Yes.
Sorry, doctor. You said that the written assignment is submitted on 12th of January. Is it by 12 or on 12th?
12 January before 12 a.m.
Means tomorrow
Okay.
night have to submit already lah. Okay.
Okay.
Thank you.
No, 12th January. No, I mean 12th January. On the 12th January, you can submit by 11 o'clock also, never mind. 11 o'clock at night is still okay. But before you enter 13th January.
Oh,
by
okay,
12th
understand.
January, you have to submit.
So
Not
means it's a
tomorrow,
Friday
you have 2
night.
days.
Friday
You
night.
have 2 days. Yes.
Good. Thank you. Bye.
Okay. Okay, everyone. That's it. Thank you. Thank you again. So we shall meet again soon. I will give you guys topic for the research project next month before you end, with 1 week before you end your final exam. Okay. Okay. Thank you, everyone. See you guys. Have a nice day. Bye. Take care. Assalamualaikum.
Okay, Waalaikumsalam. Bye, doctor.
Bye.